Bonjour, bonjour. Merci d'être nous pour encore les présentations de l'IBIS. Je suis vraiment content de voir aussi ton nom dans la semaine de relâche. Vraiment de relâche à l'IBIS. Aujourd'hui, on a un grand plaisir pour avoir une nouvelle collègue parmi nous qui va commencer bientôt ici à l'IBIS. Jullien a fait son bac et maîtrise à l'Université de McGill. Et ensuite, il est allé à Cornell pour faire un doctorat. Mais je l'avais dit que quand j'ai lu son CV, c'était quand même très intéressant pour quelqu'un qui a décliné ou refusé le bourse Vanier pour aller à Cornell. Wow, ça le fait bon. Présentement, il y a postdoc à MIT qui travaille sur dynamique des microsynthèses. Il va parler de tout. Merci.
Merci David pour me présenter. Je suis très heureuse d'être ici aujourd'hui et de donner une conférence. Et je suis encore plus heureuse de joindre le département de biologie en juin. Je suis en train de perfectionner mon français, mais pour aujourd'hui, je vais parler en anglais. So I'm going to talk about my research program on investigating the impacts of rapid satellite DNA evolution on reproductive development. So satellite DNA is long arrays of tandemly repeated sequences. And they're typically found near the centromeres of chromosomes. So for example, satellite DNA could be AAGAG repeated 90,000 times in a row, AAACTAC repeated 1,000,000 times in a row. And in the case of humans, there's a sequence called the alpha satellite, 171 base pair sequence, that's repeated 500,000 times in a row. And I'm really interested in satellite DNA because one, it doesn't code for proteins, it doesn't have really obvious functions, but almost every organism has it in its genomes in different abundances and also different sequences. And it's one of the most rapidly evolving parts of the genome. And it can make up even up to 50% of the genome. So I'm interested in what impact that has. And now, satellite DNA has sometimes been dismissed as neutral junk DNA. And now, of course, like any other part of the genome, it's subject to neutral processes, but its patterns of evolution aren't concordant with it only being neutral. And in fact, satellite DNA has been found recently to be able to co-opt a couple of functions in the cellular context. So for example, it can bind proteins that then maintain the entire genome encapsulated within a single nucleus. Satellite DNA may also have roles in transcription, which I'll get into later. But at the same time, too much satellite DNA is likely harmful, and satellite DNA has the potential to participate in activities that are generally harmful for the genome or the cell. Such as they are difficult to replicate properly, and they can often change in copy number very easily through replication slippage. They are also prone to forming various secondary structures that can then cause challenges to replication and also are prone to forming breaks. And then those breaks are really difficult to repair, especially because satellite DNA doesn't just have simply one homolog. It's often present in multiple loci in the genome. So through ectopic recombination, it can result in large-scale genome rearrangements. So the way I see it, satellite DNA has a dynamic balance between its potential to cause harm and its potential to co-opt functions. And by investigating where different processes and activities of satellite DNA fall on this scale, and also seeing how we can tip the balance, that can be the key to understanding satellites' DNA patterns of evolution, and even certain diseases we don't have the answer to yet. Now, satellite DNA was discovered over 50 years ago now, but the field has really been left behind. We still don't understand really satellite DNA's functions and activities, and that's because of a couple of technical challenges. So first, satellite DNA is difficult to study with really any type of genomics. And one of the major issues behind this is because the satellite sequence units are pretty much identical, you cannot uniquely map or align them through genome alignment or genome assembly. So as you can see here, there's no way for the algorithm to tell how these reads should be aligned. So this requires specialized approaches to study satellite DNA with genomics, some approaches which I and others have developed. And the other challenge with satellite DNA is that it can't be directly manipulated with genome editing. So typically, as a developmental biologist, in order to understand what the impact or the function of something is, we'll remove it from the genome and then assay the effect on the phenotype. But with satellite DNA, we can't target it for editing because there's so many copies of it. So you'll see one of the ways I get around that is using the natural variation that evolution has produced. So in my research program, I investigate a couple of different aspects of satellite DNA evolution, especially satellite DNA abundance evolution and chromosomal structural evolution involving satellite DNA, and try to understand the impact on developmental processes, especially mitosis, meiosis, sperm, and egg development. And to do so, I use a variety of approaches, including cytology and microscopy, genomics and bioinformatics, and Drosophila genetics. And then from what we learn on how satellite DNA impacts development, that can also go back and inform why satellite DNA evolves the way it does. So today in my talk, I'm going to give an overview of 3 of the main questions I plan to address in my lab. The first being, how does satellite DNA abundance impact development? And I'll be looking at 2 examples of satellite DNA abundance changes. And then I'll talk about how does satellite DNA instability impact chromosome segregation? And then finally, can satellite DNA have a functional role? Okay, so first I'm going to talk about how repetitive DNA has really exploded in abundance in the Drosophila virilis species group. This species group has pretty much the largest genomes in the Drosophila genus, and thus likely a lot of repetitive DNA, which you'll see is true. And what I've done here is I've done analysis of long-read genome assemblies we were able to produce. And you'll see, importantly, for the first time, we were able to identify and assemble the Y chromosome in these species. So to annotate the repeats in these genomes, I use a program called Repeat Modeler 2, which I was involved in developing back in 2020. And Repeat Modeler 2 finds both interspersed repeats in the genome, which are repeats that are present in multiple different locations. These are typically transposable elements. And it can also find more complex tandem repeats that are similar to satellite DNAs in that they have copies in tandem, but they're also present in multiple locations in the genome. And the repeat I'm going to focus on for this project is called DINE. It stands for Drosophila interspersed elements, and I'm going to be showing it in orange. So here I'm showing genome assembly annotations of these 3 species in a pie chart. The different colors of the pie are different repeat classes. And I just want you to focus here on the orange, the DINE repeat. And what you can see is that in Drosophila americana, it has more than double the amount of DINE compared to these other 2 species. And if we try to track down where that came from, here I'm showing the whole genome, not including the Y chromosome. Actually, that amount of dyne in orange is pretty similar between all three species. But if I look only at the Y chromosome, we can see now this is where the difference is coming from. And in Drosophila americana here, 70% of the Y chromosome is made up by that one repeat, whereas in Virilis and Nova Mexicana, it's 25 to 30%. So now focusing in on this Y chromosome, putting it on the phylogeny, and now I'm also sizing the pies proportional to the size of the chromosome. And what I want you to see here is in this 1 million year time span that these species diverged, a huge amount of DNA was added to this chromosome. I estimate around 15 million base pairs was added in this short 1 million year time span. And all of that 15 million base pairs can be accounted for by this single repeat here. So now I want to see how I can use this system to understand how satellite DNA expansions like this can affect the organism and development. So I have a couple ideas on what I want to look at here. I'm going to show you one of them today, and that's involving what else is on the Y chromosome. Now, the Y chromosome in Drosophila, just like in humans, is the male-associated chromosome. And in Drosophila, it contains just a handful of genes, and these genes are required to make sperm, which is a highly specialized cell that in particular has a very long tail that is required for the sperm to have mobility. And there's several genes, several proteins that go into making this tail that are on the Y chromosome. And in other species, this is from the main model species Drosophila melanogaster, it's actually been shown that repeats can end up colonizing the introns of these very important genes. And that can make the transcript associated with the gene over 1 megabase long. That is likely quite difficult to process. And in this case, this is showing that this transcript to go from exon 1 in blue all the way to the final exon in pink, it's estimated to take around 90 hours to go through this very large gene that has repeats in its introns. So going back to my system where I have this remarkable change in abundance of this repeat on this Y chromosome, I want to use both bioinformatics and in situ RNA FISH to understand what the genes compositions are in these two Y chromosomes. So for example, on this species that has a small Y chromosome, perhaps it has very normal size introns that are relatively simple to process. But then in Drosophila americana that has this huge 15 million base pair expansion, likely a lot of that expansion has ended up in the introns of the genes. And perhaps it produces these long transcripts to get through all of those repeats. And now to understand the impact of this, what I can do is I can do simple crosses in the lab to simply swap the Y chromosome reciprocally between these two species, and that will effectively change the abundance of the satellite. So remember, we can't directly manipulate it, but this is a way we can kind of get around that. And then we can go on to see what the impact that is on producing these genes from the Y chromosome. So this species here that has evolved with a smaller Y chromosome, likely it hasn't been exposed to the long introns in its evolutionary history. And so maybe when we put a Y chromosome that has these huge introns onto its Y chromosome, it will be unable to process those introns. And maybe the gene doesn't get made properly at the right amount. But then what about this species? So this species, it has had to, over the past million years, manage this huge Y chromosome. And it's still perfectly fertile, so it likely evolved ways to compensate for having this huge Y chromosome and potentially a lot of introns in its genes. So then maybe when we take those, effectively take those introns away, maybe there will be a defect in that the gene might get made too early and get degraded. So by doing this reciprocal swap of the Y chromosomes, I'll be able to evaluate both the challenges of having huge amounts of repeats on this chromosome, in addition to understanding how species evolve to manage these challenges. Okay, so now I'm going on to the second type of satellite DNA abundance change. Now, what I didn't show you before in showing you those genome assemblies is actually the bulk of repetitive DNA in this Drosophila virilis species can't actually be assembled. So here is the genome assembly pie chart I showed you before. Now I'm compressing all those colors into black, into what can actually be assembled. But in the genome, there's actually a huge amount of 7 base pair satellites that, because of inability to assemble these highly repetitive short repeats, they do not make it into the assembly. And in fact, Drosophila virilis has the highest abundance of these short repeat unit satellites of any species in the Drosophila genus. And it's dominated by this satellite I'll be showing in green, AAA-CTAC. So what I'm going to show you is how I've looked at variation within the species Drosophila virilis in its satellite DNA abundance. So this species is present pretty much all around the world. So I was able to obtain isolates that had been collected from various locations, and I did Illumina sequencing without any assembly or read mapping in order to estimate the abundance of the satellites. And here I'm just showing a summary of this AAA-CTAC satellite DNA abundance. And I became interested in this isolate here, this VER00 isolate that kind of stood up above the rest and had 15% even higher satellite DNA than the rest of the isolates. So this isolate really has the highest of the highest satellite DNA abundance. So I wanted to understand what the impact of that may be. And this time I'm going to talk about an impact on the karyotype. The karyotype is the number and arrangement of chromosomes. And in Drosophila, the way we typically look at the karyotype is we dissect the brain out of the larval stage and we simply stain it with a DNA stain. And in standard Drosophila virilis, it's maintained the same karyotype across 50 million years of evolution. And this is what it looks like. There's 5 pairs of rod chromosomes. So the centromere is at one end of the chromosome. I've marked that with an asterisk. And if we go and paint the satellite DNAs onto this picture, you can see that this highly abundant green satellite is present really on all of the chromosomes. So next, when I looked at VER00, the isolate has the highest amount of this green satellite, I found something surprising. And actually, it has a striking chromosome instability phenotype. So I found 3 different karyotype versions in this single genotype. This one on the left I call Y-fuse. It's a fusion between the Y chromosome and chromosome 3. So the centromeres that were at the end now with the fusion are in the middle. A similar independent thing happened between the X chromosome and chromosome 4. I call this X fuse A. And then finally, X fuse B is a completely independent chromosome fusion between the X chromosome and another autosome. And in addition to that, I also found a fourth karyotype version in this isolate that doesn't contain any chromosome fusions at all. So I just want to highlight kind of the uniqueness of finding these different chromosome fusion versions. Now, chromosome fusions have been found in wild species in the past, and their date of occurrence can be estimated. Typically, they're called evolutionary recent if they occurred somewhere between 100,000 and 1.5 million years ago. But what I found here is on a completely different time scale. I actually call it grad school recent, because in some cases, these chromosome fusions occurred within a 1-year period. So it was really possible to observe chromosome evolution almost in front of my eyes on a grad school timeline, which I think is pretty special. So I'm just going to give a overview of the reconstructed timeline of these chromosome fusions. So here is the ancestral karyotype of Drosophila virilis as the ancestral. And then the first event I can infer occurred in this VER00 lineage is this Y3 fusion. And I originally found that in an isolate that separated out in 2018. And then shortly after that, in between 2018 and 2019, I can infer that this fusion actually broke apart or had a fission. And that's when I found the VER00 no fuse isolate. But then shortly after that, in 2019, another chromosome fusion occurred, and that's the X4 fusion up here. And then finally, I found another version of this genotype that had separated out in another lab from 2016 to 2021. And I found that this version still retained that ancestral Y3 fusion here, but it had a completely new fusion, this X fuse B, that evolved along this branch. So I was really excited to see that these were the most chromosome fusions ever found in a single wild type genotype, and also the shortest timelines really ever observed to my knowledge. And now, not only is this interesting, but these types of things are also consequential. Now, the major mechanism that these chromosome fusions occur by is there are breaks near or in the satellite DNA near the centromere. And then those breaks get repaired with a non-homologous chromosome that has a similar sequence near the centromere. And then that results through the repair in the chromosomes becoming fused together. And now this type of process is associated with speciation, and it's also impactful at the molecular level in impacting chromosome dynamics at meiosis. So typically in meiosis, you have two homologs. They separate into different sex cells. But it's estimated that having a fusion chromosome can increase the chance that these homologs will fail to separate, known as non-disjunction, and this will result in some sex cells having both copies of the homolog and some sex cells not having any. So that's what I decided to look at next in these chromosome fusion versions. I recruited an undergraduate student to work with me to do a bunch of different PCRs to measure how often the X and Y chromosome are inherited together. And what we found is that in the control, there's a very low rate of this non-disjunction, the failure of the chromosomes to separate. And then in the VER00 Y fusion version, there's a slightly elevated rate of non-disjunction, but it wasn't quite significant with the numbers we used. Surprisingly, in the VER00 no fusion version, it had a significantly elevated rate of non-disjunction, even though it has the same karyotype as the standard control. But the most interesting thing we found is this VER00X fuse A. It had an extremely high rate of non-disjunction of 21%. And this is way higher than we expected that might occur just from a chromosome fusion. It's the highest non-disjunction rate ever found in a wild-type fly. And for context, the maximum non-disjunction rate you can get is actually 50%, and that's completely random segregation. And this is almost halfway to random, so it's really quite a high non-disjunction rate. So what I showed you with this system is I was initially looking at variation in satellite DNA abundance. I became interested in this isolate with the highest amount, VER00, and then I surprisingly found it has a striking phenotype of chromosome instability. And then I found some of the karyotypes, and especially one, has this extremely high non-disjunction rate. And non-disjunction, I realize, has a lot of implications for human health. So for example, if you have 4 chromosomes I'm showing here, and then if a non-disjunction occurs, that results in having the wrong number of chromosomes, known as aneuploidy. In the case of the orange chromosome, it's missing a copy. In the case of the pink, it has an extra copy. And aneuploidy is, if this occurs in sex cell development or in embryo development, this is highly associated with inviable pregnancies. And this is a major issue in human health because it's estimated that 50% of total pregnancies are inviable, and the number one cause is having the wrong number of chromosomes. If this occurs in somatic cells, aneuploidy is highly associated with cancer. And now it's not understood why these chromosome segregation errors occur so frequently. But satellite DNA is right there in the middle of it all. It's right there at or near the centromere that controls chromosome segregation. But satellite DNA, it's prone to breaks and it's prone to mediating rearrangements. It's challenging to replicate and repair. And it can sometimes even manipulate chromosome segregation. So by studying the challenges that satellite DNA might be causing in the centromere region, we might be able to get a better understanding of why chromosome segregation errors occur so frequently. And that will bring me into my next question. How does satellite DNA instability impact chromosome segregation? So I want to use this same system that I just showed you, this VER00 system, to ask that question. And there's a couple of different mechanisms I want to investigate. I'm going to explain one of them today, and that is, could this new satellite DNA arrangement that occurred through the chromosome fusion, could it be acting selfishly to manipulate segregation? And now the way I'm going to do that is to bring it into female meiosis and see how it behaves there. And there is a fundamental difference between male and female meiosis that I first want to explain. So in male meiosis, 4 daughter cells are produced, and all 4 of them will go on to produce sperm and have the chance to fertilize and make it to the next generation. Whereas in female meiosis, again, 4 daughter cells are produced, but 3 of them become polar bodies that never have the chance to go to the next generation. And only one of them goes on to become an egg and have the chance to go to the next generation. So theory in the past, as well as just a couple examples, has suggested that different satellite DNA abundances or different satellite DNA arrangements can actually manipulate this asymmetry in meiosis, such that that homologue that has this different satellite DNA can end up going to the egg or the oocyte, more than 50% of the time. So how I want to test if this might be occurring in my chromosome is I want to chromosome into female meiosis in a heterozygous state. So one homolog has the fusion and the other homolog doesn't have the fusion. And then I can simply look at the progeny and see how many have inherited the fusion. And now by Mendelian expectations, if meiosis is occurring as expected, then 50% of the progeny are going to contain the fusion and 50% are not going to contain the fusion. But if we have this chromosome behaving selfishly, then what I might find is more than 50%. For example, 75% of the progeny are inheriting this fusion, a sign of that this fusion is manipulating the chromosome division at meiosis in a selfish way. And this, in addition to explaining this female meiosis difference, it can also cause a higher rate of errors in the male meiosis that I found before. Okay. So with this VER00 system, I think it's going to be really powerful to understand how this different satellite DNA can impact chromosome segregation. But at the same time, it was a spontaneous discovery that I found in this species. And I also want to approach the question from a different angle, and I want to bring in now the main model species, Drosophila melanogaster. And in this species, we can actually easily look at the genes that might be involved in chromosome stability and especially impacting satellite DNA. So we can remove or mutate the genes and see how they impact satellite DNA, and also see if that impacts chromosome segregation. So Drosophila melanogaster is also an excellent system to study satellite DNA. It doesn't have as much satellite DNA as Drosophila virilis, but it still has about 20% of the genome made up of satellite DNA. And it's the main model system. It's really a powerhouse for genetics and has mutants available for many, many genes. And one of the genes I want to work on here is called Bloom. Bloom is a helicase that has been described to be involved in various processes in maintaining genome stability. Particularly, it facilitates replication through difficult to replicate regions. It functions in DNA break repair, and it also unwinds entanglements between chromosomes. And now we actually expect that satellite DNA is especially prone to all of these difficulties that Bloom works on. And now Bloom is an interesting gene because in humans, mutations in the Bloom gene cause a disease called Bloom syndrome. And this disease is associated with a very severe predisposition to cancer at an early age. But it's not understood what, at the molecular level, actually leads to this cancer predisposition. So maybe by looking at how satellite DNA is affected, we can get closer to the answer. So what I did next in this part of the project is I looked at chromosomal level instability in mitotic cells in Bloom mutants. So I had the pleasure to work with two summer students for this project, one an undergrad and one a high school student. And what we did together is prepared a bunch of different microscopy slides to look at the karyotypes of Bloom mutant flies. Again, we dissect out the brain from the larva, and here we're staining with 3 different satellite DNA sequences that allows us to identify the different chromosomes. So here is what the normal karyotype looks like in Drosophila melanogaster. There's 2 pairs of chromosome 2, 2 pairs of chromosome 3, a small chromosome 4, and then the X chromosome. And in the male, there'll be only 1 X chromosome and a Y. But what we found in the Bloom mitotically dividing cells, we actually found some abnormalities occurring. Especially breaks in the satellite DNA near the centromere. So in this case, chromosome 2 with the green here is broken in half. There's one half up here and another half down here. In this example, the Y chromosome that's pretty much completely made of satellite DNA, it has broken into pieces here. You can see with the pink and green, another small piece over here. It has broken. In addition to these breaks in or near the satellite DNA, I also found that sometimes in these mitotic cells that there's aneuploidy occurring in the Bloom mutant condition. So in this case, the X chromosome has 3 copies instead of the normal 2 copies. And then quantifying these abnormalities, the Bloom mutant has a much higher rate compared to the wild type and the heterozygous control. And of these abnormalities, they're mostly composed of breaks and aneuploidy, and the breaks are significantly enriched in the regions near the centromere compared to non-satellite associated regions. And aneuploidy occurs across all of the different chromosomes. So aneuploidy here is especially interesting because, as I mentioned, Bloom syndrome is a cancer-associated disease, and aneuploidy is also associated with cancer. Now we have the satellite DNA instability as well with the breaks in the satellite DNA in this Bloom condition. So I want to next find if there's a strong molecular link between the breaks and instability in the satellite DNA and the non-disjunction that could be associated with the disease phenotype. And one of the molecular links I want to investigate is actually the type of repair that can happen. And one of the types of repair is homologous recombination or HR-mediated repair. It uses a gene called RAD51. And essentially what happens is it uses homologous sequences to fill in the gaps caused by breaks. And now this process is expected to be really quite challenging for satellite DNA because it doesn't simply have a single homolog. And there's also the Bloom gene is involved in finishing up this process. So perhaps that too much of this type of repair occurring in this really difficult region of the satellite DNA could end up causing problems for chromosome segregation. So next, what I want to look at is I want to make a double mutant of the Bloom with the RAD51 and see how that impacts non-disjunction, if it gets better or if it gets worse. And then to see at the molecular level what might be mediating that, I want to look at how centromeres are forming on the chromosome, depending on if RAD51 repair is available or not, and if the centromeres are forming at the single location that they're supposed to occur at. And then finally, I want to look at the organismal phenotypes and see if the RAD51 repair could be associated with differences in survival in the Bloom context. And also flies can form tumors as well. Okay, so what I showed you so far is how satellite DNA can be involved in mediating chromosome instability. This is almost certainly going to fall on the negative side of the scale. I also showed you how I'm going to test how a satellite DNA huge change in abundance impacts development. And I expect adding 15 million base pairs is probably going to fall on the negative side of the scale as well. To finish up, I want to see if we can find something that might be on this side of the scale of a potential function that satellite DNA might have. And that will bring me into my final question. Can satellite DNA have a functional role through being transcribed? So now there's a couple of different contexts that satellite DNA has been found to be transcribed in. One, I talked about it a bit earlier, it can sometimes be transcribed from the introns of Y chromosome genes. Sometimes satellite DNA can form short regulatory RNAs that go back and maintain its own chromatin. Sometimes it's thought that satellite DNA can just be transcribed randomly. Sometimes polymerases will randomly fall on parts of the genome and satellite DNA could get transcribed in that random fashion. But what I think I found here that I'm going to show you next is something that doesn't fit any of these models that have been shown before. So going back to Drosophila virilis, I looked at RNA sequencing data from the species ovaries. And what I found was that this satellite AAA-CTAC in the DNA form, AAA-CUAC in the RNA form, this was actually almost the most highly transcribed repeat in the ovaries. It was much higher transcribed than even retrotransposons that have a known RNA intermediate. And not only was it highly transcribed, it's also strikingly unidirectionally transcribed. So 97% of the reads were all coming from one strand that I arbitrarily am calling the plus strand. So it really doesn't look like it's a random transcription. And then if I try to look at this transcription in the developmental context, here is the Drosophila ovary development progresses in this direction here. And what I find is that this RNA gets produced in a very specific location and timing in this ovary development. And what's special here in this group of cells, there's 16 cells in this group, and actually only one of the 16 will go on to become the oocyte, and the other 15 will just be support cells that support the oocyte but don't make it to the next generation. And strikingly, I'm finding that this RNA is found only in that cell that will go on to become the oocyte, which is really, really quite striking. So I'm interested to find if this satellite RNA might be doing something useful for oocyte development. So how I want to investigate this further, first I want to take an evolutionary approach and look at other species that have the satellite DNA. There's at least 3 other species that have the same satellite DNA sequence. I want to see if they have the satellite RNA coming up in the ovaries as well in the same pattern or not. And then I want to see if I can knock down the expression of the transcript and assay how that impacts the oocyte development. So with RNA, even satellite RNA, I can still use traditional RNA manipulating RNA knockdown procedures. So I plan to look at how knocking down the RNA impacts the oocyte development. Okay, so in summary, I showed you how I'm going to test how a huge explosion in satellite DNA abundance impacts sperm development. I showed you how I'm going to test how satellite DNA can be involved in chromosomal instability. And finally, I showed you how a transcript of a satellite RNA might be involved in oocyte development. And just to finish off, I want to highlight some potential collaboration opportunities here in EBIS. So I would be really excited to work on genomic projects in a variety of species. A lot of different groups are sequencing the genomes of many different species, and a lot of these genomes contain repeats. So I am going to be happy to help with annotating the repeats in various different genomes. I know I only talked about Drosophila today, but I have analyzed the repeats in various other species' genomes. In addition, I'm really excited to work on evolutionary cell biology and also working closely with the microscopic core as well. And I'm also really excited to bring in Drosophila as a model organism. I think it's going to be great, especially for teaching and because it's such an amazing genetic model system. And please, I'm starting in June, and I'll be looking for students to work, either an undergraduate student or a part-time student to work in the probably in the fall. So please do let me know if you have students that might be a good fit for working with me. And with that, I'll thank the groups that I worked with, both my PhD lab with Andy Clark and my postdoc lab with Yukiko Yamashita. Especially thank the trainees that I had the opportunities to work with, who have always added energy to my projects, and of course funding. And with that, I'll take any questions.